I think we pretty much worked through this whole slide set, but I wanted to go back into it just for a few minutes and start here with this idea of a movement of water in the soil once you start thinking about the plant taking up the water and how that influences water movement toward the plant. And so soil pore spacings have a big impact on that. How big are those spaces? Soil composition, of course, factors into that because that will create different pore spaces. And so here they're trying to show that the central pore region, it's easy for water to get taken up from that region. But the water that starts to cling to the soil particles, that's tougher and tougher for the plant to get access to because of the surface tension forces. And so when you think about this idea as the root is taking up the water, you actually get a tension being generated on the water in the soil. The water is all connected. They're all bonded through adhesion or cohesion forces. You have this, they refer to it as bulk flow. You have bulk flow of water through the soil toward the plant due to tension and a tension gradient. Tension is negative pressure. So this idea of pulling back on a plunger filled with water is a good way to think about it. The negative scale is down in the bottom picture there. If you haven't thought about the negative scale in a while. And so understanding where it is less negative on the negative scale. So when someone says it's less negative value, they mean it's closer to 0. So -1 is certainly less negative than -8. Okay, so you haven't thought about that for a minute. You gotta kind of refresh your brain on that. But basically, when you look at when a plant is involved now at taking up water, these parts of the water potential start to become something you have to consider for the typical plant. Now, if this was a giant redwood, we would also include size of G for gravity forces. You know, I was thinking about that. But usually we just limit it to the size of P and the size of S components. So we work through this saying that you have the size of P is your pressure due to water, hydrostatic pressure. And then size of S is referring to any solutes that you dissolve into, in this case, water. And as soon as you dissolve even just one molecule of something into water, you get a negative size of S. Okay. Now in the soil, a typical soil, so this doesn't, so I just modified this. I added this last section here, so you might want to write that down or type it in. So in typical soils, they see that the size of S, when they measure for that, it's close to 0. So they just kind of say, yeah, that's good enough. But agricultural soils can be a little different beast because of it's so intensively managed by fertilizer inputs that if you have a long-standing conventional ag soil, you might have to think about size of S more carefully and realize that that might play into it. So, but for a typical soil, they tend to say, let's just kind of forget about size of S and let's really focus in on the size of P. So when they start looking at, okay, the roots are taking up the water, what's happening to the forces on the water in the soil? And so as the root takes the water, you have this surface tension, the tension as the water is leaving the macropores quite readily, but then the micropores have to get siphoned of water, and that can happen. And then that last image on the far right is trying to show, okay, now we just have like a film of water all coating the soil particles, so they're adhering to the soil particles. That water is a little tougher to remove, and you get these, as a result, you get these curved, what they call the meniscus disc or the menisci for plural. And that is actually what develops the tension. It's this curvature at the air-water interface that puts a tension on those water molecules. And so it can be described by this equation where your size of P is -2T divided by R, and T is just the surface tension of water. And then R is that radius of the air-water interface. It's the radius of that curvature. And if you get really small a small radius. If you put a small number into the denominator, you get a big overall number. And so what this is saying is as you get a really small radius, you're going to get a really negative size of P, which means it's tension. That's tension. That's the definition of tension, negative pressure. And so you get a good negative pressure or tension right at the roots. And so if you go out further away from the roots, then the tension is less on the water. So then you get a gradient. If you have this, more negative to less negative idea, that's a gradient. And we know that water will flow. By definition, water is going to flow from less negative to more negative water potential. All right, so psi sub P is a part of figuring out the water potential. And so psi sub W, the overall water potential, is also less negative out in the bulk soil. And it's more negative closer to the root. And so it's a nice way to think about water movement in the soil. It's due to this tension gradient. And it's due to the roots. It's really the roots that kick that off and create that tension by taking up the water around it. Okay. Questions on soil, water movement? Well, I encourage you to look this over several times. Usually water potential, for most people, that takes a little bit of looking at and thinking about before you start to feel comfortable with these concepts. So we're going to continue on with these concepts into the next phase, which is, okay, now that the plant took up the water, then what? And so we left off here last time with the idea that the goal is to get the water into the xylem so that the water can get distributed to the plant. Obviously, some of the water will have to get diverted into different tissues and cells because plants need water to survive just like we do. But it's surprising how much water actually exits out of the leaf. It's the lion's share of what it brings in, it puts out through the stomata. And so some of it gets, like I said, does get diverted, but most of it actually gets moved out of the plant. And so that's the next slide set now, where we are looking at transpiration, water in the plant, which is by definition, that's transpiration. And so, it took them a long time to figure this one out. Like, how is water, especially when you start thinking about giant trees, how the heck does water move up that distance? And they can see that ATP or significant energy is not involved in getting this to happen. So then, how would this go through the plant? And so they had to do a lot of measurements. So they were really curious about the xylem, the tracheids and vessels, and looking at the design of those. Luckily, they're all empty. They're dead at maturity, so there's no cytoplasm to worry about. You don't have that kind of impact on the flow of water. It's just these empty tubes, essentially. These tracheids and vessels are empty, hollow tubes. So that allows for better water flow, for sure. And then the bigger the diameter, typically the more rapid the flow. And so vessel elements are a little bit more efficient. But remember, xylem is tracheids and vessels. And they are linked end to end to end, and they just form this long conduit for flow of water and minerals. So the dissolved minerals are brought in, of course, with the soil water. And we'll talk more about minerals in a little bit. So they finally did enough experimental measurements and calculations and experiments, and they came up with this theory, a cohesion tension theory. And so they named it at least something that makes sense. Cohesion is this idea of water molecules like to stick to each other, they cohere to each other. And then tension, again, now we're thinking this idea of size of P, that's your hydrostatic pressure or tension idea. So there's something going on with that. Here, you're looking at a leaf on the left in cross section, and they're showing the internal aspects of the leaf in more detail. You've seen something like this already, drawing. And so you have the palisade parenchyma cells, you have the spongy parenchyma or mesophyll cells, and then you can see that there's some air spaces in here, which is important because what we find is that when water exits out of the leaf, it's in the form of water vapor. It's gaseous in the gaseous state. So we have this evaporation going on inside the leaf from liquid water to vaporous condition. And then you see this little blue squiggle line out. The squiggle, anytime you see squiggle, that usually means there's some kind of resistance or frictional forces involved here. And that's what they're showing there. So they're showing the stomata are open and that the water vapor can exit the leaf. And then if you look on this far right picture here, you're seeing kind of analogous to what's going on in the soil particles. So if you kind of replace the soil particle idea with like the parenchyma cells inside the leaf, so that's what you're seeing here, there's some parenchyma cells. And then you see the blue, they're trying to draw that you have a film of water clinging to the cell walls of the parenchyma cells in the leaf. And then that liquid water starts to evaporate into the gaseous state, and then it exits out of the leaf. So in order for transpiration to happen for the plant, you have to have the stomata open. Otherwise, you can't get water movement out of the leaf. And if you can't get water movement out of the leaf, you're not going to have transpiration. And so what they say is that it actually originates up in the leaf tissue, this idea of transpiration. And so here you're looking at stomata. Figure A here is the top two guard cells with the pore in the middle. You can see how the pore is more open there. And then the lower picture with the guard cells, you can see that the pore is pretty well closed down. And so if you have the stomata open, like top image, what happened was that you had water rushing into those guard cells, and that actually causes the guard cells to move away from each other due to that trigger pressure pushing against the cell membrane and cell wall of those guard cells. That causes the guard cells to kind of move away or pucker away from each other, and that opens the pore. And then to get the pore to close, you just have the water exit out of the guard cells, and they go flaccid, or they lose that trigger pressure and they start to close over the pore. All right, so for transpiration to happen, you got to have the stomata open. So for typical plants that are just out living their life, the sun comes up, the sun strikes their body, and they'll start to open up their stomata. If everything's going okay, they'll go ahead and do that. And then as the stomata open up, then you can have the water inside the leaf start to evaporate and get into the gaseous state, and then it starts to exit out of the leaf through the stomata. So let's go back to that image. So here you're looking at again, the film of water on the cell walls of the, clinging to all the cells inside the leaf. And what we see happening here is that if you look carefully, just like what we saw with that soil particle picture, you see that get these curvatures again of the radius of the air-water interface. You start to get these curves. So those are the menisci. And what they're trying to show here, in this lower image down here, they're trying to show you differences in the size of the radius. So if the radius is 0.5 or 0.05 or 0.01, what happens to your size of P or your hydrostatic pressure if you have those different radii? And so the table over here is showing the calculation. if you put the radius measurement in and do the calculation, you can see that your psi sub p, hydrostatic pressure, so that's your psi sub p value. You can see that as your radius of the curvature gets smaller and smaller, you get this nice negative pressure, bigger, negative 15 all the way at 0.01. So that's a good tension force. Tension, tension is negative pressure. So that negative 15, that's a whopping good tension force. And so as you get the liquid water receding into tighter spaces in the leaf like that, you get those smaller radii. And basically what they did is they used this equation that I typed in over here to calculate the hydrostatic pressure, the psi sub p. So this picture on the far right here is saying the same thing, and then they've included, some images here of, the liquid water continues to evaporate inside the leaf, gets into the gaseous state. That causes the liquid water to recede into tighter and tighter spaces. So then the radius of the curvature of the air-water interface gets smaller and smaller and smaller. And the end result of that is you get this really nice tension, really good strong tension, which is negative pressure. So basically what you see in the soil in terms of when the plant starts taking up water from the soil, and we talked about that tension gradient, that's what we see happening in the plant too, in the xylem. And it's all starting up in the leaves. And so this was actually the opposite of what they thought was going to happen. They actually predicted initially that water was going to get pushed up the plant. There'd be some kind of positive pressure pushing it up. But when they did all the calculations and went and did actual took data of real life transpiration, they found that wasn't the case. It was the exact opposite. It's literally the plant is sucking water up, and it's like you have a straw in the leaf, and that's and sucking the water up, just like if you use a straw to drink pop or soda. And so that kind of idea, so it was the exact opposite of what they thought was going on. But that's the fun of science, is you get to get surprised. All right. So basically what we see, you gotta have the stomata open. Otherwise this just won't happen. There will be no tension generated without the stomata being opened. And so then the water vapor gets out of the leaf, and then the liquid water starts to recede into smaller spaces. And that causes the radius of the curvature of the air-water interface of the meniscus to get smaller and smaller. And if you put that into the equation, it actually generates a really negative number, which means it's a good, strong tension or negative pressure. And so you get this good, strong tension in the leaf. And as long as all those liquid water molecules stay connected, it is felt, believe it or not, all the way down the xylem, all the way down the tree trunk, the stem, all the way down through it. And what you have down in the roots, the further you get away, you have a lower and lower or a less strong tension. And so again, in the xylem, you get a tension gradient, really strong tension up in the leaf, lower tension further away from it, and then the lowest is in the root area, the xylem there. And so you get this tension gradient, and so it's literally like sucking the water up the plant. Yeah. Up. Yep. The higher you go, the more negative the psi sub p. Absolutely. And so with the cohesion tension theory, again, this idea of bulk flow, so when we talk about this in the plant world, we're often thinking about water molecules all being connected. Like there's no air gaps or air bubbles in the way. It's just the solid connection of water molecules. And then they can move via bulk flow, like en masse. They'll just flow up the plant through the xylem, through the xylem all the way to the leaf through a tension gradient. And the other thing that they noted is that this is unidirectional. So when a plant takes up water, it will start at the roots, take it from the soil water, get it into the roots, get it into the xylem, and then It goes all the way through the plant body until it gets out of the leaf through the stomata. So in other words, we don't see water at the top, like in the veins of the leaf, moving down at all. We don't see that. It's always roots, stem, branches, leaves out. It's never been experimentally shown any other movement. And so it's this unidirectional flow of water. And then, of course, some is getting siphoned off for use by the plant, of course, because the cells have to stay hydrated, the tissue has to stay hydrated. But the bulk or the majority of water that's taken up by the plant is actually transpired out. And so here, this image is trying to just show you that this is based on a tension gradient. So if you look at that first column over here where it says water potential psi sub W, and then the second one is your pressure potential, you can see that as you start at the soil, you have, negative 0.2, that's not much, but then you get up into the leaf and it's more negative. So you have a stronger tension force in the leaf. And again, the overall water potential is also moving from less negative to more negative. And that's how, by laws of water potential, that's how we describe water. Water always moves from less negative water potential to more negative water potential. And the psi sub P component is making it more negative up in the leaf. It's really causing a nice tension. They've also included psi sub S and psi sub G, which often can get factored in as well, depending on the situation. You might need to consider it because it will impact your value. But sometimes they get omitted if they're minor. But like I said, if we were trying to figure this out for a giant redwood, yeah, psi sub G, we'd probably factor in at least consider it and see if it makes a difference. So certainly, this transpiration process, it was a little surprising that it's this tension gradient. And then they started doing experiments of, what might influence how quickly a plant does transpiration. And that can have certainly an impact on the overall growth of the plant and how well it does overall. And so I like to think about these influences similar to how your life experience is. Like if I made you run a marathon and then I stuck you in a room that was absolutely still air, or then I put you in a room that was super windy, and then think about when would you start to have the sweat evaporate off you? Under what conditions would you have the sweat just clinging to you versus going to come off you readily and feel cool? And so if you kind of use those analogies and think about some of these, I think it helps. So like with wind, if you're sweaty and you get into a wind situation, you're going to feel cooler pretty quickly. That wind is taking that moisture off of you. Well, what's really happening, and this happens to plants too, is that your boundary layer, so this little drawing that they have where they're showing this gray surface on the leaf on both the top and the bottom, that's their depiction of the boundary layer. The boundary layer is just a layer of still air that covers any solid object that's open to the atmosphere. All right, so the surface of your skin has a boundary layer, so does the leaf surface. And if you thin out that boundary layer, that means you disturb that boundary layer so that it's not still air anymore. You get them moving, get those air molecules moving. You will evaporate off your sweat, off your skin more quickly. Same with the plant. If they have a thinner boundary layer, then the water vapor exiting out of their leaf has an easier time because there's less resistance going on. Less resistance going on as you have that the water vapor exiting out of the leaf. Okay, so wind can certainly impact. If you have a windier day, you have typically faster transpiration rates, and that impacts how often then you have to water your plant or irrigate your soil so that you keep your plants hydrated. Humidity, if, again, if I made you run a marathon and then stuck you in a really humid room, 80% humidity, or I put you in a room with 40% humidity, I think, again, you'd feel the difference in how quickly the sweat leaves your body. And so we tend to think about, well, when they've done measurements inside the leaf, the relative humidity inside the leaf is roughly 100%, almost always. It's 100% inside the leaf. So then if you think about, okay, what is the plant exposed to? What kind of a day is it? Is it, 90% relative humidity outside, or, 30? And so the bigger the difference between inside the leaf to what the plant is experiencing outside of its body, that's going to increase your transpiration rate, right? And so again, using you as the analogy, if you have sweat on you, and I put you into a room that's 30% relative humidity, you'll get rid of that sweat pretty quickly, rather than if I stuck you in a room with 90% humidity. Temperature too can, yeah. Go ahead. Too humid, it depends. So you want some transpiration to happen, because that's how the plant is taking up minerals. If it's bringing in water from the soil, that means it's getting fed its minerals. So you don't want, like, if it's 100% relative humidity in the leaf and you have days upon days 90% relative humidity, they won't transpire very fast. It means they're not taking the minerals up very well. So then probably their growth is going to get delayed. So yeah, there's some impacts to that. On the other hand, if you are experiencing days of 30%, lots of lows, well, then you're watering the plant a lot. And so that's the trade-off. So, but yeah, these things are important to think about if you're growing plants, because you have to keep on top of it. And then if you have a way, like in indoor environments, you can manipulate this. You can manipulate these factors to try to optimize transpiration rates. Temperature, I think if you think about it for a sec, higher temps, you tend to sweat off faster, to a point. If for plants, if the temperature gets too high, and then usually what happens is, on days where temperature is high, that can often dry out the soil as well. So then the plant starts experiencing drought, and then they will say, you know what, I can't pull up water out of the soil. I don't want to dehydrate. If I keep my stomata open and I'm not pulling anything in, I'm going to dehydrate. And so then the goal is to shut down the stomata to try to prevent any more water loss, so you don't, dehydrate like that. And so temperature, again, you can really see the impacts on that, especially if it's prolonged high temps. You tend to link that with drought conditions as well. So, if there's been no rain, obviously. So these, with some indoor growing, you can manipulate this pretty well, and can do it all through computer control systems, fancier operations, so that you can optimize it for each plant that you're working on. Questions on this idea? Transpiration. So kind of analogous to, it's the same idea of a pressure gradient or a tension gradient. It's technically a tension gradient, negative pressure. When you think about what's going on when the roots take up the water, it's creating a tension gradient on the soil water. And then once the water gets into the plant, you're still thinking about a tension gradient. And that tension is originating up in the leaves if the stomata are open. So around here, these plants that just natively are growing here in our ecosystems, they close down their stomata at night. And so they're not doing transpiration at night. And the main reason they close down their stomata at night is that they can't do photosynthesis at night, because some of the enzymes involved in photosynthesis need light in order to function at full capacity. And so plants around here, they just like, well, there's no reason to keep my stomata open at night, because even if I pull in CO2, I can't really do anything with it. And I can't do photosynthesis. So they'll close down their stomata at night. So that means there's no transpiration happening at night around here. Now, there are some plants in other parts of the world that will actually choose to open their stomata during the night. And I'll explain more about that in a little bit. But around here, they go to sleep at night. They just chill out. Okay. Whoops. The next topic I wanted to talk about is right next door to the xylem, which is looking at phloem. All right, so remember when we look at, well, there's a nice poem I put in there too on October, the autumn colors, so you can read that. It's a very nice poem. Read that before you study, put you in a good mood. We're going to think about the next neighbor to the xylem tissue is the phloem. So remember, xylem and phloem clustered together into the vascular tissue system. And so now the phloem is being used primarily for sugar movement through the whole body of the plant. And you've seen something like this way back when we were looking at plant cells and tissues. And so phloem in angiosperms, the flowering plants, they have phloem that are called sieve tube elements. And sieve tube elements, they get stacked end to end to end to end. So they form this long conduit where it allows for sugars that are dissolved in water to flow through the sieve tube element, through the whole plant body. Now, this is not just one direction, like what we see with xylem. We said xylem is unidirectional. That's its only option. Phloem, on the other hand, it can go pretty much any direction. Any direction. And there is a method to the madness, but it can really literally go in any direction. So what we're thinking about here is when we say sugars, when a plant does photosynthesis, it's kind of out in the common literature, they'll often talk about glucose being produced. It's really not glucose. That is the sugar that is produced in photosynthesis, and then the sugar that gets transported, it's actually sucrose. Okay. And sucrose is a disaccharide. There is a glucose component to it, but what it is, glucose and fructose bonded together. So that's a disaccharide. And that is actually what gets transported through the whole plant body. And so there's all kinds of options for different sugars, but it's sucrose. We say it's a non-reducing sugar, which basically means that it is stable and it won't react with other molecules along the way as it's getting transported around the plant body. So it's just a nice little packet of energy that can be used. We say that it has a high free energy of hydrolysis, which just means when you take glucose and fructose and you break that bond, it does release a decent amount of energy that the plant can use. So that free energy of hydrolysis just means when you break that bond, the glucose and fructose, you do get a little punch of energy from that the plant can take advantage of. It's also pretty small. It's just a disaccharide. It's not this huge polysaccharide. It's just this little thing. And so it can be very mobile. So they find when they analyze the contents of the phloem, sucrose is the #1 sugar. There are other things flowing around in there, though. There can be some other sugars. This is showing some examples that they can find. And it's not every species necessarily, and it depends on the conditions. But the vast majority of any sugar, it's going to be sucrose. And then there can be some other stuff. And then there can be some little proteins and hormones. We can see hormones in the phloem. And so it's not just sugar, but we tend to focus on sugar because that's the majority of what is in the phloem. And so when we look at how does it move, it's not unidirectional. It can pretty much go in any direction, but there is a law or a method that it has to follow, and this is the idea of source to sink. source to sink. So we say that sugar moves from source to sink throughout the plant body. And so what we mean by that, if it's a source part of the plant, source tissue, we say that that is a net exporter of sugar or it's a net producer. What that means is it makes enough sugar that it can meet its own needs, but then there's some left over. So then it can get sent and help out some other part of the plant body that's sink. And so we typically think about mature leaves as an excellent example of source tissue. Sink is just the opposite. It's a net importer of sugar or a net consumer. And so if you think about it, I'm sure you could come up with these quite quickly on your own. Roots would be a great example because roots are buried in the soil. Typical roots are buried in the soil. They're not going to be doing photosynthesis. So they really depend on the source tissue to supply them. But it could also be little baby leaves that aren't quite there yet in terms of they're not mature. They can't do it all on their own. Maybe it's a beautiful flower that's getting made. They can't do it all on their own either. So there's other parts of the body besides roots that can be sink tissue for sure. And so when they get that sugar, they might use it just like how we use sugar through glycolysis to get our energy needs met in our body. So they might go through respiration similar to what we do. They might use that sugar to grow and develop, build more cells, build more tissues. They might store it up and save for a rainy day. So you're seeing a depiction of a potato plant here where the leaves would be the source and the tuber forming underground, those would be sink. Those are really good examples of source and sink tissue. Okay, so the sugar moves from source to sink. Now they had to figure out how. How is this happening? And Dr. Munch proposed the hypothesis of the pressure flow model. So again, we got to think about pressure. So SISA P is coming into play here. And so we have, oh boy, I tell you. Okay. So Dr. Munch proposed that there's going to be a pressure gradient from source to sink. So when you think about things moving and it's primarily based off of size to P, it's going to be a bulk flow phenomenon. Bulk flow, you think about a pressure gradient of some sort when we start talking about bulk flow processes. So we're thinking about hydrostatic pressure that will develop and create some kind of pressure gradient that will allow for movement from source to sink. And it can be, the leaves sending sugar to the roots, so that's, very straight downward. It could be a leaf on one side of the tree canopy giving sugar to a leaf over on the other side, so lateral movement of sugar across the canopy, for example. It can be upward. You can have leaves giving sugar to developing fruit above it or flowers above it. And so it can be any direction. It just has to follow this source to sink pressure gradient. And so they also see that it's linked to water that is right next door to the phloem, the xylem water. And so the plant will use the xylem water to create this pressure gradient and then get the sugar to flow from source to sink. Okay, so let's think about leaves, and they're busy doing photosynthesis. They're making a bunch of sugar, sucrose. So the red balls are the sucrose. So what they're showing here is that you have the leaf cells, so the source cell here, that's your leaf parenchyma cell, doing a bunch of photosynthesis, making sucrose, and then you can see that it's delivering the sucrose to the phloem sieve tube element. Okay, so remember now, anytime you put sugar into water, you're driving your psi sub S more negative. More solutes, more negative. In this case, it's more sugar, more negative psi sub F. More sugar, more negative psi sub F. Okay, so if you get your psi sub S component to be pretty negative, that is going to make your overall water potential pretty negative. And we know that water moves from a water potential that's less negative. We'll look at over here in the xylem. Negative 0.8 is less negative than negative 1.1. So water will move from the xylem in this case over into the sieve tube element in response to that sugar dissolving in there. Okay, well, if you start stuffing a bunch of water into the sieve tube element, that's going to influence your psi sub P. Your psi sub P is going to increase. And so you see 0.6 for your psi sub P. So it's kind of this domino effect, like playing dominoes where you make them tip and they bump into each other type thing. So when you have the sugar getting loaded, you have your psi sub S getting more negative. More sugar, more negative psi sub S. That's driving your overall water potential more negative. Well, water moves from less negative to more negative. So now in response to all that sugar getting loaded in, you're going to have water in response from less negative to more negative. That puts your psi sub P nicely positive. It builds up hydrostatic pressure to get this positive pressure. All those water molecules pushing against the sieve tube element. It gets your psi sub P up. Okay. Make sense so far? Okay, so what is happening in the source is you end up developing a nice positive pressure in the sieve tube element, in the source. Now you have to think about what's happening in the sink sieve tube element. It's kind of the opposite situation now. So if you imagine this sink cell as your potato tuber, you're trying to grow lovely potatoes, okay? So now the potato tuber is like, hey, I need that sugar. I want to build this lovely big potato tuber that we can make French fries out of, right? And so you see this sugar coming out of the sieve tube element going into the potato tuber. Well, now you're getting less and less sugar there. So that means less sugar, less negative psi sub S. So you see your psi sub S is negative, but it's not so bad. Well, if you have a less negative psi sub S, that means your overall water potential is less negative. And if you compare it to the neighboring xylem, you can see that it's less negative than the xylem water potential. We know that water moves from less negative to more negative. So now water is exiting out of the sieve tube element. Well, if you have water leaving that area, your psi sub P goes down. You don't have much pressure. So here you see a psi sub P of 0.3. It's there, there's still some there, but it's not dramatic. And the big thing is, it's not the same pressure as what we find in the source that's feeding that sink. So if you compare the psi sub P at the source, it's 0.6 megapascals. You compare down where the potato tuber is growing, and you see it's 0.3 megapascal. So there's your pressure gradient. You have a higher positive pressure in the source, a lower positive pressure in the sink sieve tube element. So as long as you have a gradient like that, it's going to go, the sugar will dissolve from that higher pressure area to the lower pressure. It'll move from the higher pressure to the lower pressure area, from source to sink. Yes. So so with that, make it so also So think about salts and sugars as a solute. Just call them solutes. Like, so that psi sub s is talking, the S stands for solute in the psi sub s idea there. And salts can be a solute. Sugar can be a solute. No. Other questions on sugar movement? Yeah, this takes a while to sink in. So please spend some time looking over this material. Pardon? Which one? What did I say? Oh, sink in. Oh, no, that was unintentional. that's my tired brain not even picking up on that. Yeah, I should have taken credit for it.